Hello, welcome to Biology, the scientific study of life. Ribozymes. The idea of a catalytic role for snRNA arose from the discovery of ribozymes, RNA molecules that function as enzymes. In some organisms, RNA splicing can occur without proteins or additional RNA molecules. The intron RNA functions as a ribozyme and catalyzes its own excision. For example, in the protozoan tetrahymena, self-splicing occurs in the production of ribosomal RNA, R small RNA, a component of the organism's ribosomes. The pre-rRNA, R small RNA, actually removes its own introns. The fact that RNA is single-stranded plays an important role in allowing certain RNA molecules to function as ribozymes. Notice also a region of an RNA molecule may base pair with a complementary region elsewhere in the same molecule, thus imparting specific structure to the RNA molecule as a whole. Also, some of the bases contain functional groups that may participate in catalysis, just as a specific shape of an enzymatic protein and the functional groups on its amino acid side. Chains allow the protein to function as a catalyst. The structure of some RNA molecules allows them to function as catalysts too. The discovery of ribozymes rendered absolute the belief that all biological catalysts were proteins. Okay, notice this. RNA transcript pre-mRNA. Notice exon, intron, exon 1, exon 2. The roles of snRNPS and spliceomes in pre-mRNA splicing. The diagram shows only a portion of the pre-mRNA transcript. Additional introns and exons lie downstream from the ones pictured here. Notice that one stitch or one closer look. Small nuclear ribonucleoproteins snRNPS and other proteins form a molecular complex called a spliceosome on a pre-mRNA containing exons and introns. Notice too also closer look within the spliceosome and snRNA containing exons and introns. Notice also too within the spliceosome snRNA base pair with nucleotides at specific site along the intron. Also notice the first step or closer look or first step here. The RNA transcript is cut, releasing the intron and at the same time splicing the exon together. The spliceosome then comes apart, releasing spliced mRNA, which now contains only exon. Notice that. The functional and evolutionary importance of introns. What are the biological functions of introns and RNA splicing? One idea is that introns play regulatory roles in the cell. At least some introns contain sequences that control gene activity in some way, and the splicing process itself is necessary for the passage of mRNA from the nucleus to the cytoplasm. One consequences of the presence of exons and introns in genes is that a single gene can encode more one kind of polypeptide. A number of genes are known to give rise to two or more different polypeptides, depending on which segments are treated as exons during RNA processing. This is called alternative RNA splicing. For example, 6 differences in fruit flies are largely due to differences in how males and females splice the RNA transcribed from certain genes. Early results from the Human Genome Project suggests that alternative RNA splicing may be one reason humans can get along with a relatively small number of genes, not quite twice as many as a fruit fly. Because of alternative splicing, the number of different proteins products an organism can produce is much greater than its number of genes. Proteins often have a modular architecture consisting of discrete structural and functional regions called domains. One domain of an enzymatic protein, for instance, might include the active site, while another might attach the protein to a cellular membrane. In many cases, different exons code for the different domains of a protein. The presence of introns in a gene may facilitate the evolution of new and potentially useful proteins as a result of process known as exon shuffling. Introns increase the probability of potential beneficial crossing over between the exons of alleles simply by providing more terrain for crossovers without interrupting coding sequences. We can also imagine the equational mixing and matching of exons between completely different non-allelic genes. Exon shuffling of either sort could lead to new protein with novel combinations of functions, while most of the shuffling would result in non-beneficial changes. Occasionally, a beneficial variant might arise. Okay, back to gene. Notice that DNA, exon 1, intron, exon 2, intron, exon 3, this represents gene transcription, and closer look notes that RNA processing, translation, domain 3, domain 1, domain 2, domain 3, polypeptide, or these represent polypeptide. Correspondence between exons and protein domains in a large number of genes. Different exons encode separate domains of the protein product. Okay, thanks for listening.